ReactLab software tools have been developed to enable the detailed characterization of chemical reaction mechanisms by the global analysis of information-rich multi-wavelength spectroscopic data. ReactLab Global Analysis allows the complete determination of the reaction mechanism and yields all the species information and fundamental parameters necessary for modeling a process, a prerequisite for optimizing reaction conditions or product yields under different conditions. ReactLab therefore has a role in both academic and commercial research laboratories. The ReactLab suite currently includes two applications: ReactLab Kinetics for analyzing kinetic or time-dependent measurements, such as might be collected from a stop flow experiment or following manual mixing of reagents using a standard spectrophotometer, and ReactLab Equilibria, used for analyzing chemical titration data collected during volumetric or pH-metered experiments. In both cases, the aim is to determine the underlying reaction mechanism in terms of all the participating species concentration distributions and spectra, and computing all rate and equilibrium constants for their interconversion. This results in a complete mechanistic model of the system, which can be used for process prediction and optimization, as well as providing a robust definition of the reaction that can be utilized by others or incorporated into future work. Here we see examples of kinetic absorption spectra and equilibrium titration spectra as they appear in the ReactLab GUI. Note that ReactLab is able to analyze a wide range of data types, including UV visible absorbance, infrared, Raman, ESR, CD, and fluorescence. It is particularly suited for dealing with data from modern PDA or CCD detector-based instruments. Globalization of the analysis is the key to the unique capability that ReactLab offers. In other words, it uses the complete multi-wavelength dataset during the analysis. Such multivariate measurements are essential to fully determine mechanisms of any complexity and define the reaction parameters and species characteristics. The results include the spectra of all the species, and these really help confirm the likelihood of minor intermediates and the mechanism as a whole. Excellent graphical visualization tools in ReactLab allow deviations of the experimental data from the model to be inspected in any dimension, as well as via surface error plots. ReactLab applications are based on a hybrid MATLAB Excel architecture. All data, models and results are stored in portable Excel workbooks, and a robust compiled MATLAB analysis kernel interacts with the Excel to carry out the analysis. Flexible 2D and 3D graphics aid visualization, and Excel charting tools allow customized displays of the results. ReactLab analysis is model-based. In other words, a reaction model is provided which is believed to represent the underlying chemical mechanism, and this is fitted to the experimental data. Whether the model is appropriate will be reflected in the quality of fit results, and if this is poor, a revision of the model may well be required. The model is defined in symbolic reaction notation in a dedicated panel in a ReactLab Excel worksheet. This simple Michaelis-Menten type enzyme mechanism is entered as shown here. On this slide, we see the absorption measurements from a metal-ligand complexation titration and the underlying chemical reaction mechanism we wish to fit to the data. This quite complex mechanism can be straightforwardly fitted using ReactLab Equilibria. The model is provided to ReactLab as can be seen here, and the fit results return to the workbook during the analysis. ReactLab itself also provides flexible graphic displays to inspect the fit results in great detail. Here we see individual predicted absorption profiles at specific wavelengths displayed as a function of the changing pH, along with the residuals. Next to this is a best fit spectrum as a function of titration addition. The lower graphs display the calculated concentration distributions as a function of pH, alongside the spectra of all the coloured intermediates. The experiment itself is carried out on a stop flow. This kinetic instrument is designed to enable the rapid mixing of chemical reagents to initiate a reaction, which are then injected into an observation cell, where the progress of the subsequent reaction may be observed spectrophotometrically as a function of time. In this example, we mix a solution of acidified nickel and an ammonia solution and collect absorption spectra over the wavelength range 500 to 700 nanometers at time intervals of 0.05 seconds. The data are transferred to the data worksheet of the ReactLab Kinetics Excel workbook. The reaction mechanism is shown here and comprises a number of linked equilibria. This mechanism is transcribed into the main worksheet of the workbook. Note, the metal ligand equilibria comprise chemically observable forward and reverse rates, which are defined individually, whilst the ligand protonation and buffer equilibria are effectively instantaneous and defined by equilibrium constants. We are interested in determining the metal ligand binding rates alongside the other parameters which are previously known from other experimental or literature sources. Here we have entered the known parameters and estimates of the unknown rates. We can see how well these model the measurements as they are. In the ReactLab GUI, we can see the fit is not good from the deviation in the residual surface and the negative excursion in one of the species spectra. We can now improve these parameters by performing a fit from these starting values. Clicking the Fit button results in a perfect fit. We can now see all the spectra are positive, and we now have a random residual surface. The optimized parameters and their standard deviations are also returned to the spreadsheet. All the results, including the concentration profiles and the spectra, are also written out to the results sheet of the workbook for further processing in Excel. Having completed this analysis, the ReactLab Excel workbook contains a complete and portable snapshot of the whole session. Who should use ReactLab? There are many areas where ReactLab Global Analysis, along with its modelling and simulation capabilities, can be applied. These are a few examples, but there are many more, and the usefulness of ReactLab software in both theoretical modelling and the teaching and understanding of chemical processes and kinetics should not be overlooked. For further information, please visit our website or send us an email. Thank you for listening.